Brian from the HIV databases in Los Alamos, and I think most of you know me by now. There's a picture of me and a picture of our homepage. These should be clickable links where you can get to our database, but it's hiv.lanl.gov. Today I'm going to be covering multiple sequence alignment, really focusing on HIV-1, which has some interesting properties, and also we have some tools for it. I'm mostly going to be covering our gene cutter tool, but also talking about other tips and tricks. HIV is a particular challenge because it has more insertions and deletions than most other organisms have. I mean, I've aligned lots of other proteins and genes over the years, and HIV really has some real tricks in it. Insertions and deletions make multiple alignment hard because if you don't have any insertions or deletions, there's nothing to it. You just the input is going to be aligned already pretty much. With POL gene, protease RT region, which is now highly sequenced in HIV, there aren't very many insertions and deletions. So the protease RT region is really, really simple to align. It's pretty much like any other organism. The envelope is where things get really tricky, and then in the LTR and the end of GAG and a few other places, there are also some unique, difficult to align regions. But I'll be talking mostly about envelope because that's the really hard part to align, and I'll show you some special tricks there. HIV also has multiple overlapping reading frames, like where GAG and POL overlap and where VIF runs into envelope and so on. And then in one little piece of envelope, there's a triple reading frame where the second exon of TAT and REV are also being encoded where envelope is being encoded. So if you want to align in the TAT reading frame, you align it differently than you do if it's in the envelope reading frame. Anyway, we've made some special tools for aligning HIV genomes, and I'll give you some tips on dealing with other places where things don't work out perfectly. I won't be covering short read alignment, like using bow tie and things like that to get your short reads aligned to a single genome, but that's a separate kind of problem and topic. Pairwise alignment is really simple and fast and easy. You can use BLAST or Needleman-Wunsch or Waterman, or there's a really quick scriptable tool called Align Zero that you can put into a script to do a pairwise alignment. So it's really pretty quick to align each of your short reads or longer reads to, say, the HXB2 reference genome using Align Zero. And that pairwise alignment can be very useful to you for mapping where your sequence hits the HIV genome, or for showing where your sequence has an insertion or deletion relative to HIV reference sequence. Multiple alignment is more tricky because after you've got several sequences aligned, you have to decide where to gap the next sequence relative to the other ones. In the lower left corner here, I'm showing a typical output of a mapped alignment of HIV, and on the right is output from gene cutter of the same region, or not exactly the same sequences, but the same region of the genome. And you can see on the left that we have some single nucleotides and some pairs of nucleotides and some single gaps. And what that does is it makes it so that you can't translate this to amino acid sequence easily. Most alignment viewer programs, you can just click a button to flip back and forth between amino acid view and nucleotide view. But if your sequences aren't aligned in frame with gaps of 3 for a full codon, then it doesn't translate. So our gene cutter tool focuses specifically on getting all of the HIV genes aligned in frame. So when you view it with a alignment editor, right here I'm showing AliView views of the alignment, when you can flip back and forth between protein and DNA views. So this is just a map of the HIV genome to refresh your memory about where the overlaps are. GAG-POL is an interesting place because it's not a new start codon for the pol gene. What happens is the ribosome is coming along the message, and when it gets to a series of 6 T's followed by a stem loop structure, it can slip back and slip into the pol reading frame, and you make a GAG pol fusion protein. So for every transcript, you make some GAG and some GAG pol. So you're making about twice as much P24. Assuming it slips back 50% of the time, you would make twice as much P24 as you make P51RT. And I don't know about the ratio of slippage, but I think it's fairly constant through time in HIV and fairly constant for different subtypes of HIV, but I'm not positive if that's been proven. Then with VIF, VPR, TAT, REV, those are from spliced messages. So the first messages that come out are actually spliced because they go through the spliceosome and everything, and you make TAT and REV, and then REV binds to the REV responsive element in ONV and causes unspliced RNA to be pumped out of the nucleus into the cytoplasm. Anyway, these overlapping reading frames and multiple reading frames in a single site make alignment a little bit tricky, and our gene cutter tool will give you TAT and REV spliced, and will give you VIF and essentially give you this region where VIF overlaps with pol. It'll give you that twice, once aligned in the VIF frame and once aligned with the pol. Gene cutter has an option for how many codons between compensating insertions and deletions are allowed to be misaligned, because occasionally you'll have a real genome where there's a single insert in one site, and then a little bit further you'll have a single deletion that compensates for it. That could be a viable virus that just has now a unique coding region there. In the example I'm showing here, there's more of these slippages than you would expect in normal DNA. We usually see none of these in a genome, but occasionally we'll see one. But anyway, with the program set to allow these changes, these slippages, you can see that in the top frame there's some obvious misalignment. When I set that down to 0 codons allowed in the bottom section, you can see that I have a lot of single base insertions and deletions, but I have a much better alignment. But anyway, that's adjustable in the program. For most purposes, MAFT and other multiple alignment tools are the best option. They're faster than gene cutter. They can be scripted in. Gene cutter requires you to interact with the website, enter your email address to get the result mailed back to you, and so on. So programs like MAFT or Muscle or Clustal W, Clustal Omega, those do a really good alignment, really. They just won't be in frame. So they're not good specifically for the topic I was just talking about, getting translated to amino acids. But usually the output from those alignments are really good enough for eyeballing the data and spotting any errors or anything. I'll show you some examples in a minute. And then if you like what you're seeing with MAFT, then you can resubmit that alignment into a program like Gene Cutter that's going to take longer to get the more optimal alignment. And it's always good to use a quick alignment first and then eyeball the data. You can often spot problems and fix those problems before you send your data into something like Gene Cutter that's going to take a while. And I've tried a lot of other tools on the web. There are many servers out there where you can supposedly get your gene or genome aligned in frame. And for some of those, it can take up to a month to get your results back. This is a fairly large alignment, thousands of sequences. But, I'm waiting a month for my results. I get my results back at the end of the month, and it's like, dang, that's not any better than what I sent in. So it's really worthwhile to make sure that what you're sending in the first place is good, and also test drive some of these on a fairly small data set before you wait a month for results. Here I'm just showing a pixel tool, which is a good way to view an alignment. You can set it to be each base to be one base or amino acid to be 1 pixel on your screen, or 2 pixels by 2 pixels, or 3 pixels by 3 pixels, or whatever, to get a quick view of an alignment, very compressed. So you can see more on your screen than you would see when the program is showing you each base. Here I'm just zoomed in on the same thing. So here on the left I have a unaligned data set, and on the right it's aligned, and you can see right off the bat that when it's unaligned, the data looks pretty good. You can see that those are all going to be alignable just by eyeballing this in pixel. And on the right you can see that it got aligned, but there's some obvious problems, like right up here, that's probably 2 sequences, and you can see with the deep pink there that what happened was something unique. In each of these cases where I have a single misaligned segment, it's this pink stuff, and down here you see that that's a run of ends. So my program that I used here did not treat the ends nicely and sort of misalign them. So I could make a choice to either go in and it'd be pretty simple to fix this alignment by hand to move those ends over, or I could decide maybe I want to delete those sequences out of the data set before I align it, or I could change the ends to real bases, maybe go back and look at the output of the sequencing run by eyeball and correct the ends to the bases that are the best call for those sites. In the HIV envelope region, we have a special case in the variable loops tend to be glycosylated, and they tend to vary in the number of glycosylation sites. And whether or not you want to align the glycosylation sites to each other is a question, depending on what you want to know about the glycosylation of envelope. So we've made a special tool for that called N-glycosite, which will take an alignment like I'm showing you here, where the ends are not specifically aligned. Really what's been aligned there is envelope in general, and you can see the cysteines that form the base of the variable loops are perfectly aligned, as are sites around those cysteines. And this is typical of HIV envelope, is that there are some absolutely conserved regions. It's not all hypervariable, but then there are these sites of insertion deletion that are extra rich in glycosylation sites. And for those of you that don't know, a glycosylation site is an asparagine followed by any amino acid except proline, and then followed by a serine or threonine, and it's the serine or threonine that signals the glycosylation protein to add sugars onto the asparagine. Anyway, you could do a lot of work to try and align these glycosylation sites by hand. There's no tool that's really going to do that for you, but the glycosite tool can give you an analysis of number of glycosylation sites and type of glycosylation sites and everything without them being perfectly aligned. It will also output the alignment of amino acids with the ends that are glycosylated or potentially glycosylated, recoded as O. O is a accepted universal signal for a glycosylation site now. So anyway, in this case, I've tried to align the central glycosylation site within the variable V1 loop, just to show you what that might look like. And here you can see with the ends recoded, ends followed by S or T, recoded as O, makes it easier to realign this by eyeball to get the O's lined up if you wanted to. So here's, if I go back here, you'll see that I have 4 highly conserved glycosylation sites in this region. And after realigning the ends in the center, now it appears that I have 5. There's 1, 2, 3, 4, 5 sites because I've kind of cheated to align one more site in the middle. So anyway, depending on how you align your glycosylation sites, you can get slightly different outputs here. But in general, we have a lot of sites that just kind of shift around, and they're not truly homologous. So for phylogeny, you don't want to necessarily align these. You're better off to strip this whole region out of your alignment for getting an accurate phylogeny. AliView, BioEdit, and other multiple sequence alignment editor tools also allow you to view your alignments in different ways to highlight more quickly differences from a consensus, differences from majority, differences from a single sequence, and that really quickly makes it easy for you to spot places that are misaligned. It doesn't help you a lot to align by hand, but it does help a lot to spot the places that you need to work on. Here I've highlighted a single sequence that's a recombinant and showing you how that looks. So here we're seeing a lot of difference between my query sequence, a lot of differences from A6 here, very similar to A6 in this whole region. And then below, I can see that it's different from A6 here and more similar to B over in this region. So this is an AB recombinant. Sometimes sites don't stand out to the eyeball for being misaligned, but they'll come up in a similarity plot or other tools. So it's always good to pay attention to your data, and also think about using other tools to look at your data to be able to spot places that are potentially misaligned. And don't always assume when you see something funky in a plot like this that it's real hypermutation or something. Go back and check your alignment to make sure that there wasn't something that could just be shifted over to realign that region. A common problem with all alignment programs is this. You get a single site, the CCA, that's aligned differently in different sequences. Here the CCA is on the right. Here the CCA was pushed over to the left. I don't know why programs do this, but they all consistently will make this kind of mistake, and that's a real pain in the butt. It's fairly easy to fix it if your alignment program, which most of them do, will allow you to sort on this column. So I'll sort on either the left column here or the right column, and it'll bring all of those that are aligned one way together. Then I can change them to be aligned the other way. And then most programs also allow you then to resort back by sequence name. So having your sequence names be meaningful or logical helps you a lot when you're doing alignments, because you can, if they're named by subtype, for example, you can sort by subtype. Here's an example where most of the alignment was pretty good, but then there's this set of sequences that were kind of misaligned in just one little region. And I don't want to go back and realign my whole set here. This might have taken, a day to align with GeneCutter or something. I don't want to resubmit it, especially if that tool, GeneCutter, was the one that made this error. It's going to make the same error again. So a lot of tools like AliView allow you to select a block and just realign that one block. That's very useful. So here's the same thing realigned now. The trouble is AliView and most other programs won't pay attention to the reading frame. So you can see over here that this gap of 3 is not the same as this gap of 3. And if you eyeball it, you can see I've got a codon, another codon, then 2 bases. So I'd really like to shift this G to the left. So that's fairly easy. If you only got a few sites like this, it's pretty easy to just do that by hand. It would be nice if the tool would allow you to translate to protein first, do the alignment of the block, and then come back, but it doesn't do that. It gives you an error message. It says if you want to align nucleotides as translated amino acids, you have to realign everything. And I don't want to realign everything in this because I have complete genomes here. So anyway, that's not available in AliView yet, but that's the sort of thing that when I notice things like that, I'll write to the software developers and see if I can get them to add another feature to their tool. We've gotten the AliView people to add quite a few nice features to their editor over the years, and this is the next one I'll probably ask them to do. So anyway, perfecting a huge alignment can be a never-ending task. You know, if you start looking at individual codons and moving things around by hand, you could, spend weeks or months on a single alignment. And it's really important to decide when to just call it good enough or to consider other methods such as gap stripping, which would remove that entire V1 region out of your alignment. You know, if you want the most accurate phylogeny, you'll never get that V1 funky region aligned correctly, so it's just noise in your data. But it's really quick and easy to delete that region, either by selecting it by hand or by using a gap stripping program to strip out regions that are that have gaps in X percentage of your sequences. And that just removes that noisy region from your data. Anyway, I'm trying to keep this kind of quick and short for a webinar. And if you have any questions, you can write to me. And I'd like you to, if you have any questions now that I can answer, let's go over that before I sign off.